Well, I'd like to first thank the organizers for the opportunity to present at this meeting. My name is André Marette. I'm a researcher at Laval University. And today I'm going to talk about new mechanisms by which yogurt intake may reduce the incidence of obesity, type 2 diabetes, and fatty liver disease. I've been working a long time on the mechanisms of insulin resistance leading to type 2 diabetes. And of course, this is well known now to be associated with fat cell hypertrophy, this metabolic inflammation developing into the adipose tissue, eventually spreading to the liver and skeletal muscle. Inflammation is also known to spread to other tissues, cardiovascular tissues, and even the beta cells in the pancreas, leading to type 2 diabetes and cardiovascular disease. We came to realize many years ago that the intestine or the gut is also very important. Of course, the gut produces hormones and neurofactors that are important to regulate metabolic homeostasis, but also the gut is an important barrier to bacteria and bacterial products. And if that barrier is leaky, which we call gut leakiness, then it favors the appearance in the circulation of LPS and other inflammatory molecules, and that creates metabolic inflammation. Of course, I'm going to talk today about the microbiota because the microbiota or dysbiosis of this microbiota can favor the development of metabolic inflammation by favoring gut leakiness. So we've been working on different food factors that are known to regulate the gut microbiota. And today I'm going to focus on fermented dairy products because, of course, this is the topic of my presentation. What about fermented dairy products? First of all, there's a lot of systematic reviews, meta-analysis that have shown clearly that in general, dairy products, the intake of dairy products is at least neutral or even favorable in order to reduce the incidence of cardiovascular related clinical outcomes. And for example, in this publication by Drouin-Chartier et al. In 2016, when you actually review all the literature looking at the association between the intake of dairy product and clinical outcomes, what appears to be very, very clear is that yogurt intake is associated with a favorable outcome, meaning less incidence of type 2 diabetes, and the association is very strong, very high. So why would yogurt intake reduce the incidence of type 2 diabetes? So several years ago, we started a collaboration with Danone Nutricia, and those two individuals, Noémie Daniel and René Tonashbar, in my lab, were involved in this. They're the first two co-first authors of this paper. And their work was to determine the effect of yogurt intake on the whole body and tissue-specific insulin resistance in a new model of type 2 diabetes that we have developed. And this is a mouse model. I'm going to show you the details soon. And of course, they wanted to look at the potential role of the gut microbiota and specific metabolites in yogurt that could explain its health effects. So we designed together with Danone these diets for these animals. We had three groups of animals. We had control animals on a low-fat, low sucrose diet, high-fat, high sucrose diet fed animals, and some of those animals were also treated with yogurt. So this was a prevention trial in the animals. And we had three diets. We had the low-fat, low sucrose, high-fat, high sucrose, and in this particular diet, we introduced the LYP. LYP is a lyophilized yogurt product because it allows us to concentrate the product and put it in the diet. So the diet of these animals here contain the yogurt product, which allowed us to give the animals the equivalent of about 2 portions of yogurt per day, which is in humans what is needed to see some beneficial effect based on meta-analysis. So we kept these animals for 12 weeks on the diet. We did 3 different studies, and at the end we could pull the analysis and looking at the effect of the yogurt treatment on metabolic homeostasis and liver disease. So this is the results of the pool analysis of all these studies. So the numbers of animals is about 80 here. It's very, very high because we pulled the analysis. And you can see that as compared to control animals, the high-fat feeding produced an increase in body weight. and this was slightly but significantly reduced by the yogurt treatment, and this is probably linked to a small reduction of food intake or energy intake in these animals. So if you do individual studies, you won't be able to see this very small effect, but if you combine the studies from 3 batches, you actually see a significant effect. But even more striking is the fact that our new diet, which is containing different type of protein, is really increasing fasting glucose. So these animals are diabetic, but this effect is reduced in animals treated with yogurt. And fasting hyperinsulinemia is also prevented to some extent by yogurt treatment. And when you actually look at the OMA index, which is an index of insulin resistance, you can see that the yogurt treatment for the time of the experiment was able to reduce insulin resistance. Now, this is the individual study data for glucose tolerance. So you see the glucose tolerance of the animals on the high-fat-fed diet is really exacerbated as compared to control animals. The yogurt effect is very small. It's not significant on glucose tolerance. But when you actually look at the insulin release during the glucose tolerance, you see a major reduction in the amount of insulin that is released. And this is really the beta cell release of the insulin that is reduced because C-peptide levels are also reduced in these animals. So to confirm that this was an improvement of insulin sensitivity in these animals, we perform hyperinsulinemic erglycemic clamps. So the amount of glucose that is disposed during the test reflects the insulin sensitivity of the animal. Obviously, the control animals on low fat, low sucrose are very insulin sensitive, but the animals on the high-fat diet are clearly insulin resistant. And those that were also treated with yogurt, as you can see, yogurt prevented about 10 to 15%, so an improvement of about 10-15% of glucose disposal during the clamp. So all body insulin sensitivity is improved, but during the clamp, we also use 3D glucose to look at glucose turnover, measuring both RA, or hepatic glucose production, and RD, which is peripheral glucose uptake. And as you can see in the high-fat-fed animals, the ability of insulin during the clamp to reduce glucose production or to stimulate glucose uptake was markedly altered. This is denoting insulin resistance in both the liver and the muscle. And when you treat with yogurt, the animals that also consume yogurt, they have a slight improvement of hepatic insulin sensitivity, as shown here, but also the lack of glucose uptake during the clamp that was seen in the high-fat-fed animals was improved in the animals treated with yogurt. So indicating that insulin sensitivity in both the liver and muscle was improved by yogurt treatment. Very strikingly as well, when we start looking at liver in more detail, this is the histology of liver, you can see that the amount of lipids in the liver is of course increased by feeding a high-fat, high-sucrose diet. This is the liver weight, but this is, when you look at liver weight, you look at glycogen and water, not only lipids. So if you measure lipid triglycerides, you see that there is clearly hepatic steatosis in the high-fat-fed animals. But the yogurt treatment, particularly on lipid accumulation, really reduced hepatic steatosis in these animals. You can see that at the histology. And we had 2 pathologists that looked at these slides independently and blindly, and they confirmed that this was associated also with the reduction of signs of NASH and NAFLD when animals were treated with yogurt as compared to no yogurt treatment. We then look at the gut microbiota because our hypothesis was that yogurt increases or improve metabolic homeostasis to change in the gut microbiota. And so these are the 3 independent studies comparing here the control versus high-fat-fed animals. And you see that there's a lot of change. So red denotes a reduction of bacteria, different species of bacteria, or I should say genera. And in green, you have the increase in some species. So, of course, there's a lot of change with high-fat feeding. But what is more interesting is when you start comparing the yogurt versus the high-fat-fed animals. These animals are exactly the same diet, except, of course, that some of the protein especially was replaced by the yogurt treatment. And as you can see, some bacteria like this Streptococcus was increased by yogurt treatment. This is probably one of the starting bacteria that was present in the yogurt. You see Osteobacter that seems also to be increased, at least in 2 studies. And then the most striking results was this one, this Streptococci was clearly downregulated by yogurt treatment in all the different studies that we did. It's a non-culture bacteria. We don't know yet what it is exactly. We're looking into it right now, but it's very interesting. But not only at the taxonomic level did we see an impact. When we start looking at bile acids, we look at several type of bile acids in this particular project. And one very interesting one is HDCA, because this is associated with reduced hepatic steatosis in mice and improvement in blood glucose in LDL receptor knockout mice. And a decrease in this particular bile acid was reported as the main bile acid alteration in a rat model of hypertensive NAFLD. And this particular bile acid was actually reduced by high-fat feeding in both protocols where we looked at bile acids, and this was partly prevented. This loss was partly prevented by yogurt treatment, which is kind of interesting, and it might maybe explain some of the effect we have seen on the gut microbiota. Now, to demonstrate the causal effect of the microbiome into the yogurt effect, we transferred the gut microbiota from animals treated with the high-fat diet plus or minus yogurt to germ-free mice, producing mice colonized with the microbiota from these different groups. And when we look at the impact of these microbiota transfer to these animals, what we notice is that even if it's a smaller number of animals and only 9 weeks of treatment or follow-up after the transplantation, what we found is that already glucose seems to be a little bit improved by yogurt treatment, but not significantly. Not by yogurt treatment, of course, by the transfer of the gut microbiota from yogurt-treated animals. But clearly the fecal microbiota transplantation was able to improve fasting insulin, and the OMA-ER was almost significantly different in these animals. And when we look at glucose tolerance, we saw no difference between the two groups, as we've seen with yogurt treatment. But when we actually look at the insulin release during the test, we found out that those animals that were transferred a microbiota from yogurt-treated animals had actually lower insulinemia throughout the process. So this shows that some of the benefits of yogurt treatment were actually carried out or explained by changing the gut microbiota. Next, we wanted to compare the yogurt and liver metabolomes in order to try to reveal new markers of yogurt health effects. So when we look at the liver metabolomes of these animals, we identified 35 metabolites that were differently abundant to the liver of yogurt-treated animals versus non-yogurt-treated controls. And 6 of these metabolites were also increased in yogurt as compared to milk, and were overrepresented in the liver of yogurt-treated high-fat fed mice. So we thought these might be very interesting metabolites. And I'm going to focus on 3 of these metabolites here, which are ICA, HMVA, and IVA. These are hydroxylated metabolites of branched-chain amino acids. So leucine, isoleucine, and valine can be metabolized to ICA, HMVA, and IVA. So we thought these were quite interesting findings. And just to give you some ideas about the potential of these metabolites to maybe play a role into the metabolic benefits of yogurt treatment, when you actually look at the levels of these molecules, so this is the level of these molecules in the liver, in the animals which are on the control diet, and the animals on the high-fat fed diet actually had lower levels of these metabolites. And yogurt treatment, as expected, because yogurt contains these metabolites, was able to reverse partly or almost fully as compared to animals non-treated with yogurt. This was measured by UPLC-MSMS. We also confirmed these results by LC-MSMS. As you can see, the reduction in the liver with the high-fat feeding, partial prevention by treatment with yogurt in these animals. But interestingly, these changes were also seen in skeletal muscle, especially for HMVA and IVA, so reduction by obesity and prevention by yogurt treatment. And even in the plasma, BCHA are detectable in the circulation of these animals. And again, obesity or high-fat feeding reduces the level of these metabolites, but this can be partially prevented by yogurt treatment. When you actually use NMR to quantify these metabolites better, you see the different distribution of these 3 different molecules in the circulation. Again, these molecules are not present in milk. They are present in yogurt, and this is the measurement of these metabolites in the 3 different yogurt product we use for the 3 studies. Now, very interestingly, these metabolites of branched-chain amino acids are associated with changes in both blood glucose, fasting blood glucose, as well as hepatic triglycerides. You can see that there's a negative relationship between the abundance of these different metabolites and hepatic steatosis and type 2 diabetes, suggesting that these metabolites are associated with the health benefits of yogurt treatment. And to determine whether these metabolites are bioactive, we done some studies in FAO hepatocytes looking at liver glucose production. And we confirmed that these BCHA are cell autonomous modulators of liver metabolism. This is hepatic glucose production measured in the basal condition or the insulin suppressed condition. Of course, insulin per se is able to suppress glucose production. But you can see there's a dose-dependent effect of these 3 BCHA to reduce hepatic glucose production, especially in the presence of insulin. Also, you can show individual effect of these BCHA. For example, ICA is able to reduce hepatic glucose production, again, both in the basal, in the insulin-stimulated or suppressed state. Leucine, which is a precursor of ICA, is not able to do that, so it's really specific for the hydroxylated metabolites of BCAA. And when you combine leucine and ICA, you don't see any change, meaning that leucine cannot interfere with the ability of ICA to reduce glucose production. So this paper was recently published in Nature Communication, again, the work of Noémie and Rato, but also several individuals in my lab and at Danone Nutrition Research. And what we are currently doing is trying to understanding how this yogurt treatment can actually replenish or restore BCHA levels. So it's possible, of course, that by providing BCHA directly into the system, this is explaining why the liver contains more BCHA after yogurt treatment, but it's also possible that change in the gut microbiota conferred by yogurt treatment is helping to metabolize BCAA into BCHA, and therefore helping the liver to contain more of these metabolites. So we're currently looking also at different species of bacteria that could be contained or added to yogurt in order to increase BCHA content in different type of livers of yogurt. Finally, a few words about humans. Is this only a phenomenon in mice? We don't believe it's the case, because when in collaboration with Claude Zagagnon at our research center, we started to look at the level of these branched-chain hydroxy acids in human plasma. And as you can see, they are detectable. We can detect all of them. I'm going to show you only one example, ICA. So we're looking here at the ICA level in obese individuals. These are massively obese, severely obese individuals. You see the BMI 40 here. So these are undergoing surgery, of course. And we have here the obese type 2 diabetes individuals. So the only difference between these two, they were matched for age and sex and weight, but one group is hyperglycemic, the other one is not. So the obese diabetic individuals had almost significantly lower amounts of ICA in their circulation. That's only looking at 8 or 9 individuals. So this is very preliminary, but it suggests it's the case that these metabolites are reduced in type 2 diabetes. And when you actually look at the ICA-leucine ratio to have a kind of a point of view about the biosynthetic pathway leading to the production of these metabolites, you see that the tendency is even stronger for a reduction. So we're currently looking at hundreds of people in order to increase the N and to look at this more properly, but these very preliminary studies are quite interesting. So I'll stop here, and I'd like to thank Noémie, Renato, and several members of my lab for this study. And of course, my colleagues at Laval University who were involved in this project, and of course, my colleagues at Danone Nutrition Research, especially Adna Kutnikova and her team, our collaborators. Several people were involved also in providing the yogurt product that we use for this study. I'd like to thank CIHR for funding, as well as Danone Nutrition for funding, especially Patrick Vega and Liliana Jimenez for funding this project. Thank you very much, and I'll be happy to answer questions.